Hello, this is Jim Brewster, Education Coordinator at FamilyTreeDNA, and today we're going to be talking about DNA testing methods. So these are going to be the ways that we actually take your DNA and we read it, we process it, and we put it into data that you can then use for your genealogy. So this is going to be a somewhat advanced topic, so make sure that you have your thinking caps on. And this presentation is most likely also going to knock your socks off, so you might want to grab an extra pair of those as well. So over here on the right is a beautiful sculpture made by yours truly of DNA. Now you probably have seen this representation of this helix structure, this double helix, these two strands that are wrapped around themselves. Well, if you were to take these strands, you were to flatten them out, stretch them out into a straight line, you would get something like this. So you can see here that these are the two strands, they're sugar phosphate backbones, and they're connected like rungs on a ladder by these nucleotides. So now all humans and all DNA is composed of 4 nucleotides that we abbreviate as A, C, T, and G. And these are the letters that are used to basically write out the blueprints of how your body functions. Now, to differentiate these two strands, we generally refer to them as the forward strand and the reverse strand, because you can see that these nucleotides always occur in a pair, and that one half of each pair is attached to each strand. So we want to make sure that we keep those separate, so we call them the forward strand and the reverse strand, based on the way that they replicate. In 2000, the human genome was finally sequenced. And when it was done so, each nucleotide was given a position. So these are sort of like coordinates on a map, so that you could know where in the genome you were talking about. Now, DNA, these nucleotides, they follow a very special kind of rule. A always pairs with T, and C always pairs with G. Each one of these chemicals is in a different chemical shape, and those shapes just happen to fit together like a ball in a glove. So they always pair together with each other. So, for example, we could see here that we have an AT pair, and here we have an AT pair. But it's a little bit different because on position 1, the A is located here on the forward strand, but on position 2, the A is located on the reverse strand. Now, because we know that A is always going to combine with T, we can actually just separate this, and then just for simplicity's sake, just say that at position 1 we have an A. All scientists in the industry, they refer to the value, whether it's an A, C, T, or G at this location, based on what the nucleotide is on the forward strand. So at position 1, you could say that it has a value of A, and at position 2, it has a value of T. So even though they are both AT pairs, they're reported differently because we're only focusing on the forward strand. So let's say that we want to focus on a specific area of DNA, because remember, DNA is composed of millions and millions of base pairs. And if we have a specific research project that we want to do, and we only need to look at a specific area of it, say we only need to look at this area right here, then we don't need to sequence the entire strand of DNA. We only need to target this one area. So first, what we do is we use a special enzyme called helicase that separates these two strands into individual ones. And this is true for any of the DNA processes that we're talking about. It splits them in half, and then it makes extra copies of them through a process called amplification, so that we can read all of those extra copies, make sure that we're getting an accurate reading of it, so that if there's maybe a slight mistake in one read, we're able to catch that by essentially being able to proof it by looking at many copies of that area. So the way that we highlight the area is we use a thing called primers. Now, primers are going to be artificial nucleotides from the reverse strand. Now, this is a very simplified version of this. In reality, they're going to be much longer, but they are designed in such a way that they are only going to fit in the unique pattern that's only found in that one area. because even though you only have those 4 letters, they appear in unique patterns in certain areas. So if you can create a complementary pattern half that will go onto that, and then you add a fluorescent dye to it, then when you're running it through the machine, it's able to pick up that fluorescent dye. You've built this primer so that you know exactly that it's going to go on position 1 and 2. And so then it's able to say, hey, start reading me right here, and then go this way and find all the values that you can. And you can even actually do the same thing on the reverse strand. You can create reverse primers to make copies of all of those reverse strands that you have through that amplification process. And that's an extra way that you're able to make sure that the results that you're getting are accurate. So now you might be wondering, okay, well, you feed it into a machine and then you say, okay, it starts here, but then like how is it actually reading this? Like, how does highlighting one specific area help for the rest of it? Well, it uses a principle called fluorescent spectroscopy. Now, I know that's a mouthful. It's a big $5 word, but don't worry about it. We're going to break it down for you. Basically, what it means is that there's a principle in chemistry where different elements and different molecules, they shine different colors when they're subjected to energy. So that's why if you put different kinds of metals in a fire, for example, the fire might burn a different color because it's fluorescing, it's changing into that brighter color momentarily in a different wavelength. And we can take advantage of that because the 4 nucleotides that we're looking at, they each fluoresce in a different color. So all we need is an energy source. So here is a accurate real world version of that energy source that they use in the lab. It is in fact a Lego ray gun. And then they shine it onto all the DNA segments that you have. So you go pew, pew, pew. And then because the energy is exciting those different nucleotides, you're going to get A, C, T, and G that are going to fluoresce in different colors. And so if we start where that fluorescent primer is and we work our way out, and we know that each location is going to be in sequential order, then we can tell what the value is at that location based on the way that it shines. So you can take a DNA fragment, because again, remember, we're not looking at the entire genome. We're only looking at a targeted area for it. And when you shine that laser over it, you can see here in this representation, and this is typically the sort of data that you would get with one of the sequence types that we're talking about. And you can see that it shines a different color on each one. Some of these little bars are higher than others because just more of those copies were picked up, but you can still see that it was read at all the locations. So this is a way that we can get good reads on those, or good data. Now, the earliest method for getting this data, for using this technique to process DNA, it was the first, at least widespread technique, was called Sanger sequencing. Now, Sanger sequencing, it used that fluorescent spectroscopy principle, and it applies it in a special kind of way. It actually uses a specialized gel. So, weirdly enough, even though it is nothing like Jell-O at all, it actually comes in this festive little cake mold here from the 70s. No, I'm just kidding, it doesn't. But it is a gel layer, and it's specialized because it has what are called capillaries. It has these special channels in it that have enzymes that will attract each specific type of nucleotide. So we take our little DNA fragments, we put them into our Jell-O mold, and then it reads the DNA fragment, and you can see that it picks up in each channel, it only marks the areas where its specific nucleotide value was picked up. So the A channel is only going to make a little tick mark here where it sees A's. And then the C only where it sees C's, and so on. So you're able to then read this, and based on what marks you see in which channel, you're able to reconstruct what that sequence is based on where that fragment starts. And this has proven to be incredibly reliable. Sanger sequencing has something like a 99.99% accuracy rate. It's very reliable. The downside to it, though, is that it can only read small fragments. It has this gel that it has to physically go through. So at a certain point, it's going to sort of lose the oomph to keep going through that cell. So it's going to be small fragments. It's going to be typically somewhere between 500 and 900 base pairs. Now, for some tests, that's perfect. That's fine. That's all you really need. But when you're talking about tests like the Big Y that study thousands and thousands of base pairs, it's not really, it's impossible to get one single read that will cover the entire coverage area for the Big Y. So instead, a technique called shotgun sequencing is used. Now, I don't particularly like this name because I'm pretty sure that when the scientists were coming up with it, there was not a car in sight, and everyone knows that you're not allowed to call shotgun until you see the car. But here are the scientists doing it anyway, so I don't know, right to your congressman. But in any case, here you have your DNA fragment, and so you can see that it's a fairly short area. And now this could go on longer than this. Obviously, it's 500 to 900 base pairs, but just for simplicity's sake, I've just shortened it. So just for illustrative purposes. So now we have this special primer, and we have designed it so that it attaches to base pair 1. So when we attach the primer, we run our laser across it, pew, pew, pew. Now we get the values that we're able to read for the whole area, and now we have our DNA fragment. Now, if the coverage area that we want to cover is bigger than the 500 to 900 pairs that can be in a single fragment, we essentially just piece it together. So we then build another primer that is in an area that we know is going to fall within the coverage area of that first fragment. And so let's say that we want to put it on base pair 20. So we design a primer specifically for that area. We build out another fragment. And then just for illustrative purposes, again, it's the same thing. We then build another primer here that we know is going to be in fragment 2. We build that out. We have another one. And then just to belabor the point, I'm going to just add one more. And so you get the idea. So now, even though you have these tiny little fragments, when you put it together, you're able to get a complete reconstructed sequence. So this takes some advanced, powerful computing methods to be able to place where it is, to be essentially able to piece all of those jigsaw pieces together so that you know that the fragments that you're sequencing are being lined up in the correct order. So not all Sanger sequencing tests use a shotgun approach to it, and it's shotgun because it just shoots out a whole bunch of random small fragments everywhere. And not all Sanger sequencing tests use this. If a Sanger sequence test only needs to cover one area, it doesn't have the need to piece together the rest of it. But with a test like the Big Y, obviously it's going to need many of these. And each test is going to require its own processing time with its own gel layer, its own sequencing time, all that sort of thing. And so if you're talking about an area that's millions and millions of base pairs long, it's not feasible to be able to use Sanger sequencing for that. So instead we use something called next generation sequencing. Now, this is built on the principles that we've already talked about, but it does it in a little bit different kind of a way. So it uses something called a flow cell. Now, what a flow cell does is it has millions and millions of tiny little wells in them that are each filled with a specific primer. Then between all of the different primers, it's able to cover a very large sequence area. So these tracks here, they have all of these wells in them. For those of you who are weirded out by lots of recurring holes in a very small thing, which I kind of am, I did not add a close-up of that. But each one of these wells has a specific primer. So instead of that gel that has an area that's designed for a specific fragment, it has lots of primers that are lots of areas throughout that entire coverage area that you want to be looking at that each have primer. So then you do the same thing. You use the shotgun sequence, you break that DNA up into a whole bunch of fragments, and then you wash those fragments over the flow cell, and the different primers each bind to their specific fragment, and then it uses a camera in the same way that the other tests use a laser to be able to look at the different colors of each nucleotide, be able to read it, so you're able to align it with something like this. And an alignment is going to look something like this here. So each, in the green here, each one of these are specific reads. So you can see that they are smaller pieces of the whole. They have overlap because, again, there are a lot of probes that are all designed to cover the area so that you get good coverage of that area, but none of them is a whole. So when you piece all of these together, you're able to see that your value here is an A at this location because you have A. Oh, what's this? You have a T? Well, see, the fact that you have so many other A's, this means that this is most likely a misalignment, meaning that that probe didn't actually connect in the right place of DNA. And that can happen sometimes. There are some portions of DNA that are called highly repetitive areas. They're where the DNA repeats itself in the same sequence over and over and over. And if you're repeating that same sequence, it's hard to be able to design a probe that is going to recognize that it's attached to one part of that repeating pattern and not another part. And that results in a misalignment. So there are some areas that naturally have lower coverage than other areas because they're harder to read. They're sometimes called noisy areas because we just can't pick up where these misalignments go. But we're able to reconstruct through all of these tests an idea of your actual DNA. We then compare that to the reference genome that's been established and is used as an industry standard. And you can see here that at this location, in the reference genome, it has a value of T. So the fact that this person has a value of A means that they have that mutation or they have that SNP. They're positive for that SNP. So next-gen sequencing is a way to quickly sequence large amounts of targeted DNA. And that's because it's processing all of those fragments at once instead of compiling them one at a time. It's also able to discover new SNPs. So even if a variant from the reference sequence has not been detected in anyone, if it's within the area that we're covering, since we're looking at the whole area and not specific markers, it's still able to pick those up and report novel variants. And the cost for this is directly related to the read depth. And now that's something that's very important to keep in mind, because you can have a next-gen sequencing or a whole genome test, which is a type of next-gen sequencing that is designed to look at the whole genome. But if it only does it with a single read, a single go-through of this, then it's going to be missing large portions of the DNA. And there's ways to compensate for that I'll talk about a little bit later on. But that is one thing to keep in mind when you're hearing about, oh, it's next-gen sequencing. Okay, but what's the read depth for it? If it has a low read depth, it's not going to provide the same amount of robust information that other tests might. So to illustrate the idea of a read depth, we're going to take a look at one of the features that we offer here. So one example of a next generation sequence test is the Big Y. Now you may have seen our SNP checker tool, and this is where you're able to look up any particular SNP. Each one of these represents a read. So these are either a forward read, meaning that it was a primer that attached to the forward strand, or a reverse read, meaning that it was a primer that attached to the reverse strand. But either way, it's able to produce the value for that location. So you can see here that there are some areas that have high coverage. So this is yours truly's result. So you can see here, for example, the reference value is T. I have a C. That means that I'm positive for that SNP. There are some other areas where it looks like I might differ from the reference value, but these are most likely just small read errors, because this is an area that has had high coverage, it has had a high read depth, but only one of those reads actually picked it up. So this is what's called a high coverage next-gen sequencing test, because we cover the targeted area up to 70 times. So that doesn't mean that all 70 of those tests are going to pick up the entire genome, but when you combine it together, you get very good coverage. There are some areas of this test that have low coverage or lower coverage. So this is an example of an area where the primers weren't able to bind as clearly, and there's some areas of the genome that do that, especially if it's an area that repeats the same pattern over and over and over. It's hard to find a primer that you know is going to accurately combine to one part of that repeating pattern and not another. So this would be an example of a low coverage test. Now, with these tests, you're looking at a large area, and they're really great at being able to pick up new mutations, because they're not targeting just a specific location. They're targeting that whole area, and they're reading everything that they can in every single read. But the thing is that depending on your research purposes, you may not actually need every single read in every single area. And that is true especially for autosomal DNA. Autosomal DNA kind of more needs a spot check. It needs specific markers throughout the genome. So you don't need to extensively test the whole genome, but you have some markers on chromosome 1, you have some on chromosome 5, you have some on the X chromosome, and so on. And if you want to test them in all of these different areas, then instead what is ideal is something called microarray testing. Now, microarrays, they work, I guess, sort of similar to next-gen sequencing in that you have a chip that has these primers on them. Only this one's a little bit different. It has what are called probes, because you're not looking to start at a location and go from there moving forward. You're only hoping to study that one area. So what you can do is you can design a probe that will attach to only the one area that you want. So here's your probe that attaches to it. So your DNA, it gets washed over the chip and it attaches to the appropriate probe. And then from there, you're able to sequence the test. So it's a spot check at specific locations. And that means that it only is able to test known locations. So the Big Y or other next-gen sequencing tests, they're able to pick up new SNPs because it's looking at all the areas that it can. With something like Family Finder, there are predefined SNPs that it wants to look for. It's predefined autosomal SNPs that it wants to look for. So it's a series of probes that will pick up each of those little guideposts, and those are what we use to compare to other populations. So that means it can't find new SNPs. So here's a version or a picture of what one of our microarray testing chips looks like. So you can see the shiny part are just all of those metallic beads that are on it that are attached to probes. The DNA sample gets washed over it. And it uses, again, our handy dandy fluorescent spectroscopy approach to be able to read what the values are along the probe at each of those locations. So now these tests, they can vary a lot in what they do and what they're good for. And the biggest thing to keep in mind is the cost that goes into it, because the cost can vary quite a bit. So first you have Sanger sequencing. Now, it is an older method. It's been around for a long time, but it still is in use, and it's really great for specific targeted areas. The downside, of course, is that you have to get a lot of Jello powder for it. Not to mention the fact that you have to mix it together and then, stick it in the fridge and let it sit. And then, if one of your tests needs like some canned fruit in it or like cottage cheese, which is gross, but if you're into that, there's just a lot of considerations. So if you're looking at a large covered area, it's not really cost effective, but it is accurate. It's incredibly accurate. But it reads those 500 to 900 base pairs, so it's really better for small targeting tests. And if you want to look at a larger area, then you have a high material cost and it's a high sequencing time. But it is still used occasionally for some things. Traditionally, it has been used for STR testing, but that is somewhat changing. You then have next-gen sequencing. And this, again, I talked about. It's our handy-dandy chip. It's a pretty beefy-looking chip, honestly. And this one, it has high throughput. That means that it's able to sequence a large amount of data in a short amount of time. And that's because it sequences the whole area all at once. It's continually washing over the entire targeted area instead of compiling individual tests together. So it's able to discover novel variants that are in those targeted regions, but it does require advanced data processing, and so that's one of the costs that go into it, because it takes really powerful computers, really powerful software to be able to piece together the thousands and thousands of little fragments of DNA, like pieces of a jigsaw puzzle. And the cost for it is related to the read depth. Now, I say that because there are some companies who are shifting to a whole genome sequencing approach or a next-gen sequencing approach for autosomal tests. And those tests, they do cover that entire area, but they have a very low read depth. They have a 1x read depth. So that means that there are going to be areas that it misses. That's sufficient for things like autosomal testing, because autosomal testing requires just that spot check, just those few areas here and there. And with a 1x read, you're going to be able to cover enough of those thousands of locations. And then lastly, you have microarray sequencing. So this one is cheaper, and I think in part it's because the chip is not as beefy. Like this chip just looks beefier. Like, yeah. But this one it's a pre-configured chip. And so these, once your configuration has been created, you can mass produce these very quickly and very cost effectively. So that makes it a really approachable test for a consumer DNA test. But it is those specific locations. And it's perfect for things like a family finder. So with the autosomal test, it just looks at it in a different kind of way, but it's still able to produce that same data. So with these, it's not necessarily that one is better than another. It's that each of them approach the same data in different kinds of way. And by the same data, I mean the same DNA sequences. It's able to process all of those and able to get you the same results. If you looked at the same area using all three of these techniques, they would be able to get you the same data. So ultimately, it comes down to cost and what your goal is with testing. So hopefully that gives you a better idea just mostly of what these terms are that you might be hearing, like whole genome testing or next-gen sequencing, or this shift to it. When you hear something like whole genome sequencing, it sounds like it's a very powerful thing that is going to get you a lot of data. But you have to pay attention to what's the read depth for it, what's the testing technique that's being used for it. And that'll give you a better idea of the breadth and scope of your test. So that's all. Hope it gives you a better idea of what's going on. Come check us out at the booth, and I hope you have a great day.